Meet Clara. She'd been gardening for 11 years. Same soil, same seeds, same water. One difference. Not a bottle from the garden center, not a specialist product. Something already sitting in her kitchen drawer. Something she almost threw away. And what happened beneath that soil changed the way she saw her entire garden. Here's the question most gardeners never ask. When a plant struggles, yellow leaves, weak stems, small fruit, we look up. We add more fertilizer. We spray. We wait. But what if the problem was never up there? What if everything that decides the strength of a plant, its resilience, its yield, its ability to survive heat and drought, is decided in a place we never look, just a few inches beneath your feet? Welcome to Rooted, where we go beneath the surface, because that's where everything actually happens. Inside a single packet of baker's yeast live millions of cells. They look dead, beige, dry, completely unremarkable. But the moment they meet warm water and a whisper of sugar, everything changes. Metabolism starts, sugar converts, enzymes fire, and into that liquid pour metabolic byproducts, vitamins, amino acids, and cellular compounds that can influence both plants and the invisible world living in your soil. That's why yeast stopped appearing in research only as a baking ingredient decades ago. Scientists now study it seriously as something far more interesting. A plant growth-promoting organism. It doesn't just feed, it signals. Research shows that certain yeasts produce or release compounds, including IAA and auxin, a plant hormone that directly influences root formation, branching, and cell growth. Other yeasts mobilize phosphate, produce compounds that improve iron availability, or help roots access nutrients already present in the soil, but previously out of reach. You're not pouring in a nutrient. You're sending a signal, and in tired, depleted soil, that signal can be the difference between a plant that barely survives and one that genuinely thrives. Clara didn't know any of this when she first tried it. She just knew her tomatoes were struggling. And she'd read somewhere, half skeptical, half curious, that yeast water might help. So she tried it. What she didn't know was that she was accidentally replicating something researchers had already documented. In a widely cited study, adding Saccharomyces cerevisiae to fertilized soil increased root biomass in tomatoes by 56% compared to the control group. Not only that, nitrogen and phosphorus uptake rose simultaneously in both roots and shoots. Even dead yeast cells showed an effect, which means it's not only the living activity that matters, it's also what the cells themselves contain. Other research with rice seedlings showed measurable root dry mass increases within 2 weeks. Studies using residual yeasts from brewing found positive effects on germination, seedling development, leaf count, and chlorophyll in tomatoes and lettuce. Yeast is not magic. It is a biological tool, and it has the numbers to prove it. Here's the part most people never hear. For a long time, we thought that roots worked like straws, passively and silently, that they simply absorbed all the dissolved nutrients in the water flowing past them. That picture was wrong. Modern research describes something far more active. Plants at their root tips don't just wait, they interact. They pull microorganisms in, extract what they need, and release them back into the soil. The microbes that survive this process exit through root hairs and trigger the root to produce even more hairs, creating fresh pathways for the next round of capture. It's a loop, a cycle, a living engine that fuels itself. This is why a living yeast solution is so different from a mineral fertilizer. You're not delivering dead salts that wash away with the next rain. You're delivering living workers. Fuel for a biological engine that once started keeps turning on its own. And once that engine starts, roots don't just grow. They surge, they branch, they hunt. Here's what Clara discovered that her neighbor didn't. She tried the same recipe, same concentration, same timing, and barely noticed a difference. Because yeast is not a universal switch, it's a signal, and the soil has to be ready to receive it. In sandy, depleted, biologically poor soils, the response tends to be strongest. The signal arrives in a system that has been waiting for exactly this kind of input. In rich, already active soils, the effect is more modest, because that system is already running well without help. This is what biostimulant research consistently emphasizes. Results depend on soil type, plant variety, concentration, timing, and climate. There is no single answer. There is only observation and adjustment. Clara's soil had been exhausted by years of the same crops, the same rows, the same routine. Her neighbors had been rebuilt with compost for a decade. Same yeast, different conversation. And understanding that difference, that's what separates a gardener who gets results from one who just follows recipes. If yeast can do all of this, why isn't it on every garden center shelf? The answer is simple and uncomfortable. Mineral fertilizers are perfect products, standardized, long shelf life, easy to ship, predictable in every climate, every season, every store. Living systems are harder. They age, they respond to temperature, they require care and precision. But here's the thing, the freshest, most effective version of this, you can make yourself, in your kitchen, in 5 minutes, for less than the price of a coffee. Not because anything is being hidden, but because living biology simply cannot be pressed into a standardized mass product the way a mineral salt can. Now the practical part. One important note first. Research works with precisely defined strains and controlled conditions. This home recipe is a biological approximation, a gardener's experiment, not a laboratory replication. Treat it with curiosity, not expectation. You need 1 liter of lukewarm water, warm to the touch, not hot. 2 tablespoons of dry baker's yeast, around 20 grams. 1 teaspoon of sugar. The sugar is not fertilizer. It is ignition. It tells the yeast, wake up, multiply, begin. Stir everything thoroughly, then cover loosely, never airtight. After 30 to 40 minutes, a soft foam cap should form on the surface. That foam is your signal. The workers are awake. Now the step that determines success or failure. Dilution. Mix this 1 liter of concentrate with 10 liters of clean water. Never apply undiluted. A concentrated solution can stress young roots and cause osmotic shock. Apply directly to the root zone. Best results with tomatoes, cucumbers, beans, peppers, zucchini, lettuce, strawberries. When? Once after establishment. Once just before flowering. Once at fruit set. And one last thing. This brew is alive. Use it within a few hours. The spark fades quickly. Treat it fresh. Use it fresh. 3 weeks after Clara first watered with her yeast solution, she noticed something. Fine white feeder roots had pushed up near the soil surface around the base of her weakest plants, like the plant had suddenly remembered how to live. By the end of that month, The plants she almost pulled out carried enough tomatoes to fill a basket. Not because of a miracle, because the invisible engine beneath them had finally been given the fuel it needed to start.